It is my distinct pleasure to welcome Lane to Morgantown and to WVCTSI and share with us some of the exciting work that he's been doing now for the last 2 decades plus. Lane obtained his undergraduate, AB, then master's, then DVM at the University of Missouri. Subsequently went to North Carolina State and obtained his PhD, stayed in North Carolina for his postdoc, went back to Missouri as an assistant professor, and over the time rose in rank and became a full tenured professor, and he's at Columbia currently, and also has an appointment in the Dalton Cardiovascular Center. Over the course of his career, he has made some seminal contribution to the epithelial transport, and unlike me, who's stuck in the gut, Lane's also worked in the airway epithelium as well as in the reproductive epithelium. His primary interest has been in terms of Cftr as well as acid-base transporters in epithelial cells, and most recently he's really got an incredibly exciting model which is an in vitro intestinal 3-dimensional model, which, as you can well imagine, really opens that expertise up to a variety of very specific questions one can ask, mechanistic questions that we can ask without having to worry about the enteric nervous system, blood flow, and everything else that goes into an in vivo model, or the limitations of a purely in vitro model with just cell cultures. So I'm very happy to have Lane come and visit us. Over the course of the time, Lane has published approximately 100 papers, book chapters, as I said, resulting in some very seminal contribution to intestinal physiology, as well as more than 100 plus abstracts. As you all know, physiological, clinical, translational relevance is all the rage in at the NIH now, but Lynn and I have been talking about physiological relevance to research for much longer than it's become fashionable. So it is my distinct pleasure to welcome Lane to speak with us.
Well, thanks, Uma. That was I really appreciate the invitation to come here and speak to your new CTSI center. Morgantown reminds me a lot of Columbia. They're both beautiful college towns, and everything seems to revolve around the university, so it seems very familiar. As Uma said, our laboratory has been interested in Cftr knockout, the intestinal portion of CF disease. And as, let's see, can you see the cursor here? Okay. And I want to convince you that the Cftr knockout mouse is a good model of this CF intestinal disease. As many of you may know, Cftr is the principal anion channel in the intestine, and it's also well known as being an important channel in the airway. And loss of this channel results in a failure to secrete salt and water by various epithelia where it's important, so you get a dehydration in the luminal content. And in fact, the patients as well as mice are refractory to secretory diarrhea. And if you look at the graph over here on the right, this is just a Nusing chamber experiment set up to do pH stat to measure bicarbonate secretion. And if you look at a comparison of wild type and Cftr knockout duodenum, follow the blue trace, which is a measure of anion secretion as indexed by the short circuit current. The green trace is bicarbonate secretion. You can see when you activate Cftr by stimulating cyclic AMP, you get an increase in current. About 3/4 of this current is chloride secretion, which is indicated by the ability to block this with a chloride uptake inhibitor bumetanide. And at the same time, you get an increase in bicarbonate secretion, which is about 1/4 of the current. And it looks delayed here, but that's simply the time it requires to titrate the solution for the bicarbonate secretion. Now, the important point of course, is down below here. As you can see, you have a lower anion secretion by carbonate secretion, and it's virtually unaffected by cyclic AMP stimulation. Another part of the disease is what is termed mucoviscidosis, which is another term for cystic fibrosis. And as you can see from this photomicrograph, the histological appearance is very similar between human Meconium ileus intestine and a CF mouse intestine. You appreciate the goblet cell hyperplasia in both. There's tenacious mucus, which is remaining within the crypt lumens here. And what you can't appreciate is there's a defect in the goblet cell degranulation. A third aspect of the disease is dysbiosis, and what that means is there's an abnormal bacterial flora. There's an increased amount of gram-negative bacteria, and this has been measured in both humans and the CF mouse. There's small intestinal bacterial overgrowth in both species, and in both species there's a dysmotility, which probably is responsible for the overgrowth, as well as deficits in the innate immunity. And shown on the right here is a study we did in the CF mouse where we looked at untreated as well as a cholite treated. So we put cholite, an osmotic laxative, in the drinking water to prevent the obstruction in the mice, and they can virtually live an entire lifespan. But what you see are these red granules within the mucus here, and even within a cholite treated mice. You still see these granules build up in the crypt lumens. Where these are coming from are the Paneth cells, which secrete these granules, which are largely antimicrobial peptides. So there's a failure of this dissolution of these peptides to modify the luminal bacteria. The disease manifestations, both humans and mice, show growth retardation. Both have been documented to have low-grade bowel inflammation. There's this obstructive disease. As you can see here, there actually was an obstruction here. It broke free in the fixation, but as you can see, it was attached through the mucus clear down into the crypts. So it's a tremendous surface area of attachment there, requiring very often surgical removal. So I kind of call them Velcro-like impactions. So we see both in human mouse the Meconium ileus as well as the distal intestinal obstructive syndrome in older patients. And this is all contained within the intestinal epithelium, because in the CF mouse they don't get airway or pancreatic disease. So it's intrinsic, if you will, to the intestine. A less well recognized aspect of the disease is an increased risk of GI cancer. I'll talk about this later. This hasn't actually been studied in the CF mice, although it has been shown there's a hyperproliferation of the intestinal epithelium in CF mice.
Is there any problem with the reproduction?
Yes, the females have problems with reproduction. The males are fertile, though. There's not the absence of the vas deferens as there is in humans. So I'm going to concentrate on first on hyperproliferation and how this relates to ion transport, and then a little bit on the obstructive bowel disease, kind of a new area for us, mainly with the a focus on this problem with mucoviscidosis. So with all this going on in the intestine, it's difficult to see what is really just intrinsic to the loss of CFTR in the epithelium. CFTR is expressed by far and almost exclusively within the epithelium of the intestine. Its highest levels are expressed in the crypt epithelium. This is, of course, the site where the stem cells as well as the transit amplifying cells give rise to the cell types which are 95% enterocytes or absorptive cells, and then the secretory lineages are the other 5% of goblet cells, endocrine cells, and these Paneth cells, as well as rare tough cells which have a kind of an unusual and less well-known function. So how can we find this out? Well, the advantage came from developmental biologists. who, in searching for stem cell markers of the intestinal epithelium, found the leucine-rich G protein-coupled receptor 5, or LGR5, as a marker of the intestinal, more rapidly turning over intestinal stem cell. And they tagged it with EGF, took a single cell, and from that put it in matrigel culture with the appropriate growth factors, and grew an intestinal organoid, which has an epithelial-lined cavity, and started growing crypts off the surface of this organoid that have virtually the same number of cells, same types of cells as found in vivo. And you don't even need to really isolate the stem cells. You can simply take isolated crypts, place them in this matrigel culture, apply the growth factors, and within a matter of days get the formation of this central organoid with crypts coming off. The nice thing is you can get thousands of these from a single mouse intestine, and they can be passaged on a weekly basis for greater than 8 months. The schematic diagram of this are that you have these crypts coming off, and they come off where the stem cells localize in the organoid. So you have crypts, as well as what they call the villus domain lining the central cavity, and what causes is responsible for the need to passage these. The cells undergo migrate up, undergo anoikis, and are sloughed off into the lumen. So this lumen starts to fill up with these apoptotic cells. So we wanted to use this as an epithelial structure. As many of you probably know, this has been a real problem with intestinal research, is the inability to have a well-differentiated primary culture. And this has eluded us until just at the end of 2009. So we engaged in a study to see if CFTR is expressed in these crypts and if it's functional. As you can see here in a study we published this year, if we compare the protein expression of CFTR between freshly isolated crypts, or primary enteroids as we call them, because they're coming from the small bowel, or passage 1 enteroids, you can see we have very nice levels of CFTR expression. CFTR knockouts are control here for the antibody. And these are actually quite high levels of CFTR. If you take the whole intestinal epithelium, which includes the villus epithelium, it's very difficult to see much CFTR because it's diluted out by the villus epithelium, which has very low levels of CFTR expression. Is CFTR functional? The organoids are bendable to microelectrode studies. We can pale the cells, as you can see here. If we have a cell impalement here, we get a nice membrane potential. These are impaled across the basolateral membrane. We get a membrane potential about minus 50 millivolts. We apply forskolin to activate CFTR, and we see what we expect, a depolarization of the cell membrane potential. And then we retract the microelectrode, and it goes back to 0. If we take another cell from the organoid sometime later, while it's still exposed to forskolin, to stimulate the cyclic AMP, and then impale another cell, apply the CFTR inhibitor 172, we see partial repolarization, indicating this is CFTR activity we're measuring. And then the proof's in the pudding between the wild type and CFTR knockout. With forskolin, we see in a number of these impalements, we see a depolarization which doesn't occur in the CFTR knockout, which remains elevated above, even in the presence of forskolin, above that of the wild type. Whoops. And then finally, is there fluid secretion? Well, we've dissected off a single crypt from an organoid, left it in the gel for a few hours, and what you're going to see is we apply a forskolin as well as carbachol cocktail to stimulate fluid secretion. And you'll see the contents of this crypt flush out and even spin around in the stream of secretion. So it hits, you get the the secretion. You can see these granules that were flushed from the lumen just continue to kind of spin around in the convection there. So we get remarkable access to these crypts. So what are the epithelial-specific effects of the CFTR knockout on ion transport? So we're looking at an intestinal epithelium that's absent of the inflammation, the microbiome, all the systemic factors, and immunity. But before I get into that, I just want to point out a few things that we've learned about CFTR in the gut. CFTR is expressed mainly in the villus, but in the duodenum you still get a pretty measurable amount of expression up on the villus. So we took advantage of that using BCECF to measure intracellular pH on intact villi from ex vivo sectioned intestine. And the typical experiment to measure what we were interested in is the effect of losing CFTR on chloride bicarbonate exchange in the villus epithelium. And as you can see, what we've done is set up conditions where we have chloride on the lumen here. When we remove chloride, what we do is run what we call in reverse mode, the exchanger in reverse mode. So chloride leaves the cell in exchange for bicarbonate, and you see the intracellular pH alkalizes. And then when we return chloride to the apical media, you see the reacidification as we run the exchanger in the forward mode. Two things we learned though while we were doing these studies. First of all, as you can appreciate from the CF trace shown up here, we had reduced chloride bicarbonate exchange. And I'll explain that in just a minute. And surprisingly, we found that the intracellular pH was more alkaline. And I think these things go towards the present model of CFTR function in the apical membrane. And that is it gets rid of bicarbonate, and it does this as a bicarbonate channel. It's a much better chloride channel, but it can secrete bicarbonate. But also by providing a way for chloride entering by chloride bicarbonate exchange to then not build up in concentration and kill the gradients. And it's recycled through CFTR, if you will, back out to the apical membrane so the anion exchanger can continue to secrete bicarbonate. And we've identified the main chloride bicarbonate exchangers in the villus epithelium. So back to the question, what happens in the crypt to CFTR knockout on cell pH? And to do these studies, we use SNARV-5F as a pH sensor and use confocal microscopy. We counterstain with quinacrine, so we identify the goblet cells as well as Paneth cells. And what we found was the CF enterocytes maintain an alkaline pH. We take stacks of images and using this 3D reconstruction program, IMERIS, we place measurement spheres confined within the borders of the cell. We can turn it around, look at it in various angles and place this measurement sphere. And what we found was that if we apply the CFTR inhibitor, the cells go alkaline, so this is an acute setting. But what was interesting that in the CFTR knockout, where they've been born without CFTR and continued on, they still show this alkaline pH in the epithelium. And so we're kind of interested in why there isn't compensation. And so we've done kind of a mini array, if you will, of acid-base transporters comparing wild type and CFTR knockout litter mates, 8 pairs. And what we found is kind of interesting. We found a number of transporters changes. We found 4, maybe 5 that were decreased in activity and 1 that was increased in activity. The one that's increased is anion exchanger 2, which is located on the basolateral membrane. And when we do functional studies like we did in the villus, we find also that there seems to be increased CFTR or basolateral chloride bicarbonate exchange activity in the CF mice when we force the anion exchanger to be functional. Now these things make sense. If you look at the things that are downregulated, they're all what result in bicarbonate loading. So the cell is responding by changing a number of transporters a little bit. And what it's doing is downregulating the bicarbonate loaders, carbonic anhydrase 9, which is important in removing CO2 or protons from the cell because it's an extracellular protein. The sodium bicarbonate cotransporter, electrogenic 1, is a main pathway for bicarbonate uptake. We found that this anion exchanger, SLC26A6, actually under basal conditions prefers to bring bicarbonate into the cell. And sodium hydrogen exchanger 2, which is a proton efflux pathway, is also downregulated. So all the bicarbonate loading or getting rid of or sustaining protons within the cell are downregulated in an effort to overcome this alkalinity. The upregulation, as I mentioned, was AE2, which should be able to handle the load. The problem is that CFTR is also a chloride channel. And it's been documented by electron microprobe analysis that there's increased amounts of chloride within the crypt epithelium in CF. And so what happens is this diminishes the gradient for chloride to enter the cell. So even though there may be more AE2 and we can force it and see an increased activity, it probably is not very functional in vivo because of this inability of CFTR to get rid of chloride via the apical membrane. Why we're interested in the alkalinity of the pH is it's well known that this facilitates cell proliferation, and this has been seen, as I mentioned, in CF mouse. It's favorable to the cell cycle, it's favorable to membrane biogenesis, and we think it may also be altering the Wnt beta-catenin signaling, which is central to maintaining the stem cell population. GI cancers, for the most part, about 80% or more have mutations in the Wnt beta-catenin signaling pathway. And this makes sense with terms of CF. There's an increased risk of digestive tract cancer in CF patients, about 6-fold increase. And this is probably the tip of the iceberg because CF patients, the average age is right now sitting around 38 years of age. So as we push forward treatment, and the average age is likely to be more important in the future. Now, I don't want to get into the whole canonical Wnt signaling pathway, but one aspect of this is that beta-catenin being important in the expression of these genes of proliferation is typically post-translationally broken down by a destruction complex. And when Wnt ligands its receptors, it causes a breakdown of this destruction complex and allows beta-catenin to increase in the cell and go to the nucleus. And one aspect of this is that it recruits disheveled to this complex, which has a positive charge domain that actually binds to the negative charges on the phospholipid bilayer. And it's been shown in Drosophila that in an alkaline environment, disheveled tends to associate more with the membrane. And so we think it might facilitate the signaling. So we've taken a look at disheveled 2 in the wild type and CFTR knockout mice. And what we see is that there's more generalized within the epithelium of the presence of disheveled 2, whereas in the CFTR knockout, we think we're actually picking up a more margin, where it's more marginated to the cell margins, if you will, and perhaps is facilitating the signaling. So this is one of the hypotheses we're investigating. For proliferation, we use a fluorescent BRDU, if you will, EDU, to measure S-phase cells. And we wanted to set our media so that it was proliferating at similar rates to that we find in vivo. So in vivo, in the duodenum, we find a wide range of proliferation within crypts, but an average of about 10 EDU positive cells per crypt. And in the wild-type enteroids with our growth medium, we also see a wide range, but our average is close to that of what's been found in vivo. Now in the growth medium, the thing that's being pushed really there is stem cell. And that's denoted by budding off of new crypts, because every bud there's a stem cell and typically a past cell associated with it. And so in the growth medium over several days' time, you see more and more crypt bud off and form. Crypts. If we remove these growth factors, which includes R-spawn and noggin and EGF, what you see in the maintenance media is that the much less budding, and what you see is usually an elongation of the crypts in the culture. So when we started looking at the CFTR knockout organoids, we thought we might be onto something, because very often we would see much longer crypts in the CF cultures. So is the intestinal proliferation epithelial specific? If we remove the epithelium from the animal and look at the proliferation, is it going to be hyped up as it is found in vivo or not? And what we found was that just like in vivo, if we look at EDU positive as a total of the number of crypts, that we see a small increase in proliferation in the growth medium. And interestingly, when we go to the maintenance medium, the CF cultures tend to maintain this rate of proliferation, whereas the wild types decrease in proliferation. So we actually get an even bigger difference in the rate of proliferation. So these indicate that proliferation is intrinsic to the CF epithelium and doesn't require any of the input of the other cell types to cause it, including the inflammation. Now we're focusing on whether CFTR is active in the stem cells. So we have mice which are expressing the Elger5 EGFP. You can see the stem cells here. It's a mosaic expression, so not every stem cell is going to show it. But we've been able to measure the intracellular pH, apply the CFTR inhibitor, and we find that there is an increased alkalinity in the stem cells when we apply the CFTR inhibitor, which is consistent with CFTR activity. And we're now collaborating with Scott Magnus at University of North Carolina. He has a SOX9 EGFP expressing mouse, so it's not a mosaic expression. And a certain fraction, the low expressing for SOX9 EFP co-segregates with the Elger5 stem cells. And as you can see, in the low expressing, even though we haven't loaded these very heavy with protein, we still see CFTR on our Western blots. So it's indicative that probably CFTR is present. And so what actually the technique now is to measure the buds, the number of buds that come off over time as a measure of stem cell activity. That's kind of illustrated here. If we really juice them up with some Wnt3a, what happens is the organoid changes shape. And actually what's going on here is a proliferation of a stem cell population. And as time goes on, what you see is just a massive budding off from this organoid, so that by day 8 here, you can see every square millimeter on this organoid is covered with a new crypt. Okay, I want to change gears here for the last part and just talk about goblet cell hyperplasia and goblet cell malfunction in CF. If you take a CF mouse off of the coli treatment, you really see a lot of goblet cells. Okay, as you can tell here, it's becomes very hyperplastic or metaplastic. But even on the coli, you can still appreciate an increased number of goblet cells being produced. And we also measure things we believe are involved in the signaling towards goblet cell differentiation. We think TLR2 and 4 contribute to an IL-13-based stimulation of this transcription factor, SPEDF, which has also been found to be increased in asthma as well as CF lung disease. And so all these things are favorable for goblet cell differentiation. So the question is, what if we isolate the epithelium now from the microbes, all the dysbiosis, the immune system? And what we find is that the goblet cell hyperplasia goes away in CF. The Villus-like domain, the Crypts, there's no difference in the number of goblet cells between CF and wild type. The signaling goes back to no different than wild type. And even if we try to stimulate goblet cell hyperplasia by applying IL-13 to the organoids, you see there's no difference really in the number of induced goblet cells between wild type and CF. So the basal and stimulated goblet cell hyperplasia does not appear to be intrinsic to the CF intestinal epithelium. However, what we have found, or what we're interested in, is this defect in goblet cell degranulation. And I could only find one report of this in human CF intestine, which is shown here, where there's a failure of the goblet cell to release its granules normally. But we've been seeing this all the time, even on mice on colite, this anchoring of the mucus to the theca of the goblet cell. And you can appreciate that in many instances, almost every cell, you can see some mucus that's tried to release and still remains attached to the theca. So this is in vivo. The enteroids certainly have goblet cells as well as pant cells, and they're functional because we can stimulate their release by the muscarinic agonist carbachol. So that brings up the question now, is this defect intrinsic to the epithelium? And the answer is yes, it is. If we look at a wild type cell, goblet cell in a wild type enteroid and stimulate with carbachol, we just see a kind of a brief refractal plume come off from the goblet cell as you see it degranulate. In CF, you still see this attachment of the mucus to the goblet cell. You can see, even appreciate a couple out of focus here in this single crypt. And just to illustrate, I've got a couple of films to show. This is a wild type. This is one side of a crypt. This is the lumen of the crypt on the right. Here's a goblet cell we're focusing on in a wild type. And if you watch, you'll be able to see the mucus coming off, I think. Maybe not. You'll see it kind of float away here, right in this area. Let me do that again, just so you can appreciate that. Okay. In CF, we've actually already started to degranulate when we did this, but right here is a goblet cell that's already started degranulating, and you can appreciate it's the slow sludgy movement out and still attached to the theca, so it just accumulates in the lumen. What type of image? This is Marsky. 60x objective. And this one I think illustrates better. It's a little out of focus because we were actually perfusing the lumen of these enteroids, trying to wash the mucus away. And but what you can see, here's the lumen, here's the crypt, and what you'll see is this degranulation where it just remains attached. Here's another one where it'll bubble up, and another one bubbles up here. Another thing we found interesting, and we're checking in vivo, if you watch this, you also see some ectopic excitosis. So some mucin is leaking across the basolateral membrane. So we're checking to see if this happens in vivo. So these things kind of suggest the CFTR's in the goblet cell if the goblet cell's screwed up. But the more recent evidence has shown me this real nice immunofluorescence coming from you et al. Shows CFTR everywhere but where there's a goblet cell. So it tends to not be there. So there's kind of the idea that the goblet cell may no longer express CFTR. So we thought this was interesting. We thought maybe we'd get a functional correlate. And using this 3D imaging software, in about 4 months of my postdoc's time, we were able to actually place a measurement sphere within the limited cytoplasm of the goblet cell. I actually think this is the first time this has been done. And then he also placed spheres in the cells immediately adjacent to the goblet cell. And this was to measure intracellular pH. Now what he found was that in the goblet cell, the CF was no different than the wild type, which we found kind of interesting, but it was the neighboring enterocytes were significantly more alkaline. Furthermore, which is maybe we need to check this further, but it seemed that in a separate study when we were doing some more high-resolution imaging and placing the measurement spheres, it appeared that actually the apical membrane tended to have a gradient between apical and basolateral in the wild type, which was missing in CF. So it tended to be more alkaline towards the apical pole. And it seems to be a standing gradient, which I'm not sure I understand. But why this is important is the most current hypothesis proposed by Paul Quinton, that when these mucin granules are packaged, they're packaged with calcium and low pH protons, and the idea is these positive charges shield the negative charges on the mucins. And then when the secretory pore forms with the outside, the idea is that not only does sodium displace some of this positive charges, but bicarbonate may be important to neutralize the protons as well as precipitate calcium. And when that happens, you get rid of the positive charges. It allows the mucin granule to expand 1,000 times by this electrostatic propulsion. So we thought that was interesting, and the fact that we had alkaline cells immediately adjacent to the goblet cell tends to fit with that idea. So we thought we'd test it. So we perfused these enteroids at high rates, 500 times the maximal secretory rate of a crypt. We put the pipettes right in front of goblet cells and perfused it for 10 minutes, and then we had fluid flowing over the top of these organoids that were gas with CO2 and had bicarbonate, and we could apply carbachol to induce compound exocytosis. And we also put a dye in there to make sure we were contained within the organoid. And when we did this, we, even with Krebs bicarbonate ringer forced against the surface of the goblet cell, we still saw a failure of this mucus to move from the goblet cell theca. We applied 5 milligrams per mil of dithiothiotol, which is it's supposed to be a mucolytic concentration. No change. It still had the CF goblet cell still had this problem. And then to test Paul's hypothesis, we used 150 millimolar sodium bicarbonate right up against the surface. We might have had some modification of this. It's not as shaggy as some, but you can see it's still attached to the theca. So what we're thinking now is that something screwed up with the goblet cell granule. And so we actually have now measured the goblet cell theca for pH using lysis sensor dye. And so this is a theca down below here, and then we have the IMERIS program place measurement spheres at the apical, middle, and basal thirds of the theca. And what we found was pretty interesting. If we did this in wild type, you can see there's no pH gradient really from basal to apical. But in the CF, it was alkaline. Alkaline at the post-secretory pathway all the way up. So I need to hurry. And this is important because there's some very important things involved in mucin granule maturation that are pH dependent. So the low pH is necessary for cleavage of the C-terminus, trimerization of the polymers, and granular organization. So in a editorial to the Quinton article, when they were talking about you had to have bicarbonate on the outside of the cells, they also mentioned the fact that there may be intracellular communication through the gap junctions. So this has kind of been important to us in our present hypothesis, where we think in the wild type that you have CFTR exiting bicarbonate, no buildup in the cell, get normal granule formation. CF, you have this failure of bicarbonate release, or it acts as a proton sink, and perhaps through the gap junction in communication with the theca. And this allows this malformation of the granule so that it can't fully expand upon release. So just in conclusion, these enteroids have been very informative, looking just what happens when CFTR is gone from the epithelium. And with using this for the CFTR knockout, we see hyperproliferation seems to be remain and is intrinsic to the epithelium, and we're associating with alkaline pH. Goblet cell hyperplasia, on the other hand, we find to be secondary to the intestinal environment, and such as the microbes and immune system, whereas the goblet cell degranulation defect appears to be intrinsic. We find it's not improved even when we perfuse over the goblet cell with these compounds. We find the mucin granules are alkaline, and we're now working on the hypothesis that the principal defect of the goblet cell may be either in the exocytotic process or this abnormal granule formation. And I'd just like to acknowledge people in my lab, my postdoc, Jinghua Liu, who's done a lot of the work with the organoids, Nancy Walker, who's expert in electrolyte transport, new graduate student, and my collaborator who helped us develop the organoid system, Akifumi Owatani, who's now in Japan. So, thank you.